Thank you very much, Chiara. I hope you all can hear us. Thank you for the opportunity to present our laboratory and the ongoing research we're carrying out here in Florence. We are gonna show a little bit of slides if I'm able to do that. And basically, we're based in Florence, as I said, and this presentation will be shared by myself and Chiara, as you already mentioned. We are located in the main campus of the University of Florence biomedical area, that is the University Hospital of Raggi. We are part of the Department of Experimental and Clinical Medicine, which is one of the largest departments of the University of Florence, which the mission of which is to promote and coordinate research activities in the field of experimental and clinical medicine. Here you can see some of the building. This is a pretty old building where you have the administration of the department, but most of the buildings of the hospital are actually newer. We have a number of resources which are available and that might be of interest for carrying out the projects that would be supervised by myself, but particularly by Dr. Raggi, who will be completing the more detailed part of this presentation. Especially, we have several tools and platforms for preclinical imaging, tissue engineering, and especially a very advanced flow cytometry lab that is part of our department and that is exploited in several collaborations with the immunology group. With other departments and with the hospital, we are sharing different facilities, like a fairly large animal facility, a bioinformatic platform for data analysis and omics data, proteomics platforms, sequencing platforms, advanced microscopy with confocal, multiphoton. This is also in collaboration with the physics department, and scanning probes for cellular imaging. This is where actually most of the biomedical research is going on. This is the biomedical experimental campus that it's called Cubo. That means like a cube, because originally it had a more cubic shape, but now it's actually much larger. It's in close connection with the hospital. And this is just a picture of some of us. You can recognize Chiara, myself, and other staff of the laboratory, together with recent foreign guests like from Turkey and from Japan, for example. The focus of our research is the pathogenetic mechanisms of liver diseases and especially hepatobiliary tumors. And because Chiara will be the mentor in, if we will be having some of the proposals, I will leave the microphone to Chiara to explain which areas are more specifically addressed during the last 2-3 years. Chiara?
Thank you, Fabio, and good afternoon to everyone. So in the last year, our major interest is the study of the steaminess in cholangiocarcinoma. And among the ongoing and the recent published project, we have 3 main areas of interest. We have the tumor metabolism, the tumor microenvironment, and also the identification of new targets. Among the tumor metabolism, we have recently published a paper on the role of the oxidative metabolism, and also we have just submitted another one on the role of the fatty acid metabolism. On the other hand, we are very, very interested in looking at the role of the infiltrating macrophages expressing a specific tyrosine kinase, which is called MERTK, and this specific subject of macrophage enhance the tumor progression. At the end, also, we are very interested in identifying new targets, and we have recently published a paper on the role of the ERK5 kinase on the molecule of SERPINB3, as well as the importance of the sonic hedgehog pathway. And we are still working on the role of the ganglioside pattern. Ganglioside are a specific class of lipids, are ganglioside, glycosphingolipids, sorry. Since we, as Fabio has already said, we are a translational group, we are working by using preclinical models, including in vitro and in vivo. Among the in vivo, we are very interested in developing three-dimensional cultural system, including spheroids with and without scaffold, but also patient-derived organoids. And here I just want to stress the fact that we are working very close to the department of the hepatobiliary surgery. On the other hand, we are using chemical induced model as well. For the in vivo part, we use chemically induced model as well as subcutaneous xenograft model. And we are in the process to develop another model by using cell injection of liver cancer cell directly into the liver by using an ultrasound, a photoacoustic guided protocol. So I think I can conclude here my overview, and thank you for your attention.